Good afternoon, everyone. It's a pleasure to be here and be again in China at the Great Wall meeting. Congratulations for this meeting so far. I'm really enjoying very much. So, like I said, my name is Gabriele. I'm a cardiologist currently based in Berlin, Germany, and I also have the pleasure to be an associate editor for Circulation together with other basic translational associated editor, including Professor Wen Zhao and many other professionals in our team. So, what I would like to do today is to stress a little bit upon what kind of basic translational paper Circulation is looking for, and what could be one of the recipe by which a paper, basic translational, can be submitted and hopefully accepted in Circulation. As has been said by Joe, Circulation is fundamentally a clinical journal. However, we strive to get the best preclinical and basic translational science space out there, based on the fundamental understanding that there is no progress in knowledge without the understanding of the fundamental mechanism of disease. And with Joe, we really strive to understand this better, and we had an interesting and important call to validate as much as possible what kind of basic translational paper can be submitted to Circulation. So this is a little roadmap. What kind of consideration should be made when submitting a paper in the realm of basic translational science to Circulation? As many other journals, first and foremost, scientific rigor is the utmost importance. Reproducibility, transparency, and proper experimental design are definitely needed in order to achieve the goal of informing and moving forward science in this direction. Again, we are a clinical journal, so the basic translational science that we publish should be rooted to cardiovascular medicine and the relevance to fundamental question, a fundamental clinical question that can be answered and perhaps towards basic translational experiment. So unresolved question in terms of clinical relevant, significantly clinical relevant conditions. Again, important reproducibility is particularly important for basic papers and translational paper. So having accurate phenotyping of preclinical model with the detailed methodology and that anybody can reproduce for is one of the fundamental things that are necessary when you are doing your research and ideally submit it to our journal. Of course, we prefer human relevant models. For example, most of us in the lab work on rodent models that are able to mimicking some but not all of the human condition. But of course, if you are more interested in or you are working on invertebrate models, for example, that are very good to understanding the fundamental basic knowledge of science, but perhaps cannot really model what kind of cardiovascular disease we see in human beings, perhaps this is not often, but there are few exceptions. But in terms of preclinical modeling, we definitely favor mammalian models in this direction. And of course, mechanisms. Mechanisms are important, and novel mechanism of disease is something that we really look forward to publish in Circulation. Of course, there are multiple ways of understanding mechanism of disease. Some of them are perhaps incremental. If this mechanism occurs in cell A, might also occur in cell B, and this might be a little bit below our threshold in terms of interest of paper, but also what is important to recognize that our mechanism, the mechanism in our paper, needs to be fundamentally novel and innovative. We also have, of course, each of us in lab use omics, and we are not publish many omics atlas paper unless you have a very unique population condition for which there is the first omics that has ever been done in that specific disease. We tend to use omics as a discovery platform for mechanistic understanding, rather just a description of the constellation of the changes that occurs in the specific condition. And finally, again, one more time, any kind of novel biology that you discover in your preclinical models, in your in vitro system, needs to have a reality check for human condition, a cardiovascular disease. As to we encourage and we accept a paper both with a top bottom approach. So you have a disease, you look at your patient, identify a specific association, a specific phenotype, and you work out the mechanism in your mouse, on your other models, but also bottom-up approach in which you discover a novel biology and you find this novel biology in the human relevant condition. In the next few minutes, I would highlight for you a few papers that check all these hallmarks and that we've published, and perhaps can inform a little bit about what kind of a basic translational paper we are publishing in Circulation. This is an example of a very prominent and important clinical question. Many of you are familiar with the checkpoint inhibitors, now are used largely in the cancer field, but they have a shortcoming that this drug can cause severe cardiomyopathies and severe myocarditis in particular. And what this investigator did, again, was to start from a fundamental clinical question: what is the cause of ICI, immune checkpoint inhibitors, myocarditis? And they started from a human population treated with different type of ICI, some that develop myocarditis, some that did not develop myocarditis, and then work out the specific immune profiling in this patient. Again, these are human, and as human goes, it stays at the level of association. A description of the human catalog, or the immune catalog, changes that occurs in ICI myocarditis. But again, what they show here was that a specific subset, a specific type of immune cell associated with a specific phenotype in human. But again, this is association. It's very important and very informative because it can help to guide clinical choices in this condition. But we care also about the mechanism. So they use preclinical models that mimic what occurs in human being and replicate the observation found in human in the mouse model, dissecting a specific mechanism by which a specific subset of immune cells in ICI myocarditis is triggering this specific condition. Another example that stems from the similar condition is, in this case, applied to heart failure, in particular heart failure with preserved ejection fraction (FPEF), and the novel emerging risk factor that many of you have heard of, which is clonal hematopoiesis. Again, two fundamental clinical problems: heart failure with preserved ejection fraction and clonal hematopoiesis. What are the mechanisms that can link these two things together? Again, starting from the clinical observation that in heart failure with preserved ejection fraction patient, there is an abundance of clonal hematopoiesis with a specific type of clonal hematopoiesis in a clinical cohort that was identified in a well-characterized heart failure patient. They then went to identify the specific mutation that was most abundant in this heart failure cohort, and then use a mouse that can mimic clonal hematopoiesis as well as heart failure with preserved ejection fraction, and ask the question: is this association that I've seen in patient is actually mechanistically and causally linked to the disease? So again, similar to the previous paper, modeling in mice what occurs in a human being and work out the mechanism was done in this condition. As you can see here, mouse with the specific mutation identified in the human population mimics the human situation, and they work out the mechanism by which the specific mutation is associated with worsening heart failure with preserved ejection fraction. And finally, as been mentioned by Joe before, we every year assign to the best basic translational paper in Circulation the Lo Scalzo Award. Last year, this went to a group from Peking University that really moved the needle towards a better understanding and better therapeutic approach of atherosclerosis using an innovative approach such as vaccination. And one important thing is that we as a associate editors in the basic translational assign the Los Award not to the paper that received the most citation, not to the paper that has been mostly downloaded from our website, but to the paper that we really think can move the needle forward to a better understanding of that specific condition. And this is what this investigator from Peking University did. So they evaluate a number of potential epitopes, a number of potential targets that can be triggered and can be targeted in atherosclerosis. These are mouse data. They identified a specific epitope that increases in atherosclerosis and in after balloon injury in mice. They found the antibody that is able to blockade this specific epitope and the antibody, and then they tested in a mouse model of both atherosclerosis as well as two models of atherosclerosis, ApoE knockout mice, as well as LDL receptor knockout mice, and they show that in mice the inhibition, the vaccination with this specific, this specific epitope reduce atherosclerosis. Again, this is mice. Rodent models are very good, but they might not mimic exactly what occurs in human being. So they move to a large animal model. So in this case, there are two preclinical models of disease in which the mechanism and the specific contribution of this vaccination has been worked out. You can see here pig with a stent in their coronary. They develop this specific model, and when this pig are treated with the antibody, again, this ADAM7 specific epitope, they develop less neointima formation and ultimately less atherosclerosis. So again, we assign this every year. This year, in 2024, went to another atherosclerosis paper, the Los Award. We don't assign Los Award only atherosclerosis paper, I can assure you. But also in this case, what this investigator did was to investigate a specific form of atherosclerosis, and in particular calcific vessel, which is a big and important condition that occurs in human beings. Calcification of arteries track with cardiovascular mortality and heart failure, but really the mechanism by which artery gets calcified are so far poorly understood. So this is an example of a paper in which the use of omics is informative to discover the mechanism of disease. In this case, they use carotid artery and aortic valve, both normal and calcified, and they were able to describe the atlas, the proteomics, as well as the extracellular vesicle content of these two types of lesion. But having said that, once you know what are the changes that occurs in the proteome of this specific disease, then you have to pinpoint the mechanism and find a specific target, a protein target that can be indeed used for further mechanistic understanding. So they integrate this analysis using network and pathway analysis in this case, and they identified a specific protein called, in this case, HTAMCs, and they work out the mechanism both in vitro or in vivo, demonstrating that the presence of this protein is indeed causal to calcification. So I will stop here, and this is just a glimpse of what we do in terms of circulation of basic translational scientists. Thank you so much for listening, and I'm happy to answer any question in the discussion. Thank you.